Hi guys, today we're going to be covering the first half of chapter 20, which is the electron transport chain. These are my learning objectives for chapter 20. You're going to need to describe the purpose of the electron transport chain and ATP synthase, how they are functionally coupled, how the process of oxidative phosphorylation is influenced by oxygen and NADH, and how cellular ATP to ADP ratio is going to regulate this. Now, we're only covering the electron transport chain. We're going to get into oxidative phosphorylation in the next lecture, but you need to know these overall objectives for chapter 20. In eukaryotic cells, aerobic processes are going to occur in the mitochondria. Glycolysis is considered an anaerobic process, and so it's going to occur in the cytosol. So in the electron transport chain, basically you have a series of carriers that transfer electrons from NADH and FADH2. And we talked about these two carriers that were generated during the Krebs cycle. And those are going to transfer those electrons to oxygen to reduce it to water. And these reactions all take place on the inner mitochondrial membrane. There are 4 membrane-bound complexes that are in that inner membrane. And then there are 2 additional mobile electron carriers called coenzyme Q and cytochrome C. And so basically what we have is a series of oxidation and reduction reactions where electrons are transferred from FADH2 and NADH to each complex until they eventually reach oxygen. Ultimately, NADH and FADH2 are oxidized and oxygen is reduced to water. What's really cool about this is that those complexes are also involved in creating a proton gradient or a pH gradient. And we're going to learn about that more in the second part of chapter 20. But this process ends up being the driving force for creating ATP, which is known as oxidative phosphorylation. And this gives rise to most of the ATP in the cell that's associated with oxidation of glucose. So in the glycolysis and the Krebs cycle, you really only end up with a few ATP molecules. But here we're going to generate tons of ATP through this process called oxidative phosphorylation. And like I said, this process of transporting electrons is the driving force behind creating this proton or pH gradient. And what you have is you have a difference in hydrogen ions between the matrix and the intermembrane space. So just for a reminder, mitochondria have an inner and an outer membrane. And all of these reactions are occurring on the inner membrane and pumping hydrogen ions into the intermembrane space, creating a high hydrogen ion concentration or a low pH. And so that's called a gradient because there's a difference on one side of the membrane versus the other. Here you see highlighted the electron transporters that are involved in creating that proton gradient. And that proton gradient is going to be coupled to ATP production. There are a couple that are not. The mobile carrier coenzyme Q and cytochrome C is not coupled. And also, additionally, FADH2 is not coupled. But the rest of them are going to be involved. Complex 1, complex 3, and complex 4 are all going to be involved in creating that proton gradient. So why do those electrons move from one molecule to another? And a useful way to look at electron transport is to consider the change in free energy associated with the movement of electrons from one carrier to another. So if we have 2 electron carriers, for example, we have NADH and coenzyme Q, are electrons more likely to be transferred from NADH to coenzyme Q or vice versa? And in reality, we need to know what the reduction potential for each carrier is in order to know which way electrons are going to move. A carrier with a high redox potential is going to be reduced if it's paired with a carrier of a lower redox potential. So how do you measure redox potential? In this figure, we see an experimental apparatus used to measure standard redox potentials for coupled carriers. So the reference point is in the half cell on the right, where hydrogen ion is in an aqueous solution in equilibrium with hydrogen gas. Reduction of hydrogen ion to hydrogen gas is considered to have a voltage of 0. So that's our reference point. The sample to be tested is in the other half cell or beaker on the left. This first image shows a sample half-cell pair measurement for the standard reduction potential of ethanol and acetaldehyde. Because electrons flow towards the reference half-cell and away from the sample half-cell, the standard reduction potential is negative, specifically negative 0.197 volts. And as you can see, the reaction occurring is the oxidation of ethanol to produce acetaldehyde. In contrast, on this next one, the fumarate-succinate couple accepts electrons from the reference half-cell. That is, reduction occurs spontaneously in the system, and the reduction potential is thus positive. For each half-cell, a half reaction describes the reaction taking place. And the reaction taking place here is the reduction of fumarate instead of actually succinate. So this is why we have a positive redox potential. So standard redox potentials for many biological half reactions have been calculated. There's a couple I want to highlight for you here. We're actually showing a bunch of them, but the first one here is the reduction of NAD to NADH. And that has a very negative redox potential, negative 0.320 volts. And then the next one I want to show you is the redox potential of oxygen. And that has an actually very high redox potential of 0.816 volts. And what you see is that electrons are donated from NAD through the electron transport chain, and ultimately the final acceptor is oxygen. So they're going to be going from a very negative redox potential to complexes that are accepting them that have ever increasingly higher or more positive redox potential. So that's what you're going to expect to see as you're looking at whether an electron will go from one compound to another. Is it's going to go from the more negative to the more positive redox potential. So we can actually calculate the delta G aught, or the free energy change of a reaction under standard conditions, if we know the redox potential of the two half reactions. So for example, you can take the ones that we used earlier for NAD and oxygen, and basically in order to find out the delta G aught, you take negative N, which is the number of moles, times Faraday's constant, times the difference in redox potential between the two half reactions. And this is going to tell you if a reaction is endergonic or exergonic. So basically, standard reduction potentials provide a basis for comparison of redox reactions. And we know the sequence, or we can figure out the sequence of electron transport chain based on using these redox potentials. So basically, we see that electrons are going to be transported from a molecule that has a low redox potential to a molecule that has a high redox potential. And what you're going to see is that the electron transport complexes are organized in more negative redox potential to more positive redox potential order. So NADH is oxidized, and it's going to reduce complex 1, which is a multi-enzyme complex, and that we'll talk about in a little bit. And that's going to donate electrons to coenzyme Q, also called the ubiquinone, and then that's going to donate electrons to complex 3 that has a higher redox potential. Complex 3 then donates to cytochrome C, which has a higher redox potential, and then cytochrome C donates to complex 4, which has an even higher redox potential. Finally, those electrons are donated to oxygen, which is reduced to water, our final electron acceptor in the electron transport chain. So let's talk about each of the complexes in the electron transport chain and what happens. So you have the first complex, which is called NADH CoQ oxidoreductase, and it's named that because that's what it does. So basically what it does, it's a multi-enzyme complex that catalyzes the transfer of electrons from NADH to coenzyme Q. And this is going to be an integral inner mitochondrial membrane protein, meaning it's fitting right there in the inner mitochondrial membrane. It's also going to include several proteins that contain iron sulfur clusters, and this flavor protein pictured here that oxidizes NADH. This flavoprotein has a flavin coenzyme molecule called flavin mononucleotide, or FMN for short. So again, let me remind you that this is a complex, so there's multiple enzymes here. But what you see is that there's a transfer of electrons from NADH to the flavin portion of the flavoprotein. And then in the next stage, FMN is reoxidized, and then the oxidized form of the iron sulfur proteins that are contained in that complex are reduced. In stage 3, what you see is that reduced iron sulfur proteins end up donating the electrons to this coenzyme Q, which ends up being the reduced or hydroquinone form. So down here I'm picturing this oxidized versus reduced quinone, or coenzyme Q. So basically the overall reaction that is catalyzed by that first complex is that NADH reduces coenzyme Q. Therefore, NADH is oxidized. And this is a highly exergonic reaction. It releases enough energy to drive phosphorylation of ADP to ATP. And you can see that in this graph below. And the delta G for this reaction is negative 81 kilojoules per mole. Complex 2, called succinate coenzyme Q oxidoreductase, catalyzes the transfer of electrons from succinate, which you find in the Krebs cycle, to coenzyme Q. So this is going to occur in 3 stages. In the first stage, succinate is oxidized to fumarate. So go back and think about the Krebs cycle and recall which reactions this occurs in. And in this reaction, you see a reduction of FAD to FADH2. The E here represents the enzyme that FAD and FADH2 are bound to. So then in the next stage, this FAD or flavin group is reoxidized, meaning it's going to give up its electrons to another component in this complex 2, and that component is an iron sulfur protein. And basically by giving up those electrons, it's going to reduce the iron sulfur protein. In stage 3, that iron sulfur protein then donates its electrons to coenzyme Q, and now we have a reduced coenzyme Q. So the overall reaction we see is that succinate plus the oxidized form of coenzyme Q produces fumarate and the reduced form of coenzyme Q. This is an exergonic reaction. However, the energy is not enough to drive ATP production, so we actually don't see any proton pumping during this step of the reactions. Next, I need to talk to you about cytochromes. So why? Well, electrons are passed from coenzyme Q to cytochromes. And what are cytochromes? Cytochromes are groups of heme-containing proteins in the electron transport chain. So heme, we've heard that term before when we talk about hemoglobin. Basically what it means is it's an iron-containing compound. And you're going to see a bunch of different types of cytochromes that are distinguished by lowercase letters such as A, B, and C, and sometimes there's a possible additional subscript like 1 or 2. So you might see C1, and that just means there are different types of cytochromes that are present in the electron transport chain. Like I said, all cytochromes contain a heme group, and there's a lot of differences in the side chain depending on the heme, but the big thing you need to know is that the heme groups are going to be in cytochromes, and iron is going to be an integral portion of those heme groups. So when we talked about complex 1 and complex 2, you'll notice that they both reduced coenzyme Q. But now that coenzyme Q is reduced, we need something that's going to oxidize it, and that would be complex 3. So complex 3 is called coenzyme Q cytochrome C oxidase. And this is going to catalyze the oxidation of the reduced coenzyme Q. And in this process, electrons are produced by this reaction are passed along to cytochrome C in a multi-step process. The overall reaction is shown below, and there's 2 molecules of cytochrome C that are required for every molecule of coenzyme Q. You can see complex 3 pictured here right in the center, but you also see complex 1 and complex 4. They're not showing complex 2, even though it is also an integral part of the inner mitochondrial membrane. It's just not involved in proton pumping, whereas the other three are involved in proton pumping. What's interesting about complex 3 is that it contains two B-type cytochromes, which are involved in the Q cycle, which I'm going to talk about in just a second. So you need to note that coenzyme Q is going to pass electrons onto complex 3, but then in order for those to continue to be passed along, they're going to need to be involved in what's called the Q cycle. So what is the Q cycle? Basically, it's a series of reactions in the electron transport chain that provides a link between two electron transports and one electron transfer. And basically, it involves the flow of electrons via cyclic path from coenzyme Q to other components in the complex. And the reason for that is that coenzyme Q can carry two electrons, whereas cytochrome C and the other cytochromes can only carry one electron at a time. So it actually depends on the fact that coenzyme Q can exist in three different forms. The oxidized form, which you see at the top, the semi-reduced form, which you see in the center, and the fully reduced form that you see on the bottom. So what you see is that one electron is passed from coenzyme Q to cytochrome C, leaving coenzyme Q in the semiquinone form or with just one electron bound to it. And you're going to see that repeatedly. That's why it's a cycle. It donates one electron at a time, even though it's going to be able to accept two electrons from the previous complex. So what you see is that complex 3 is involved in this Q cycle, and this is what's going to provide a mechanism for electrons to be transferred one at a time from coenzyme Q to cytochrome C. This complex 3 results in proton pumping, and the reactions in this complex are going to be exergonic enough to drive ATP production. Cytochrome C oxidase is complex number 4. That's going to be our final complex, and it's going to catalyze the transfer of electrons from cytochrome C to oxygen. So this is the overall reaction that you see here, and ultimately this is going to result in proton pumping as well. So this is going to be another complex that's involved in the proton pumping that you're going to see. It is an integral part of the inner mitochondrial membrane as well, and it actually contains two cytochromes as well as two copper ions that are going to bind to the electrons that are being transported. So those copper ions are intermediate electron acceptors. Basically what you see is that cytochrome C donates to this complex, and cytochrome A is the first acceptor, which then donates to those copper ions, which then donates to another cytochrome A within the complex, and finally you're going to have oxygen. That's the final electron acceptor. The reaction of the cytochromes is seen here, but what I want you to note is this overall reaction, where you see that a half O2 molecule is reduced with two electrons to produce H2O, or water. So we don't really have a half oxygen molecule floating around. So what we really have are 4 whole electrons that need to be donated in order to split that oxygen molecule and to produce 2 waters in the process. I want to show you the energetics of electron transport in this table, and what you're going to see is that the majority are going to have a very negative delta G, or they're going to be very exergonic. I want to bring you back to this figure to put everything we've learned all together. Hopefully this will make it make more sense, and then you can go back and watch the slides again if you need to. Basically what you see here is that NADH donates electrons to complex 1, and they're passed through the various enzymes in complex 1 over to coenzyme Q. Coenzyme Q is reduced, and then it is oxidized when it donates its electrons to complex 3. Recall that it can only donate them one at a time, so therefore there's a Q cycle that helps enable the donation of 2 electrons from coenzyme Q, one at a time. And then complex 3 donates the electrons to cytochrome C, and then cytochrome C donates those electrons to complex 4. Ultimately, complex 4 donates those electrons to oxygen, and what we see is that out of a half oxygen molecule, you will get 1 water. So in the end, you're actually going to have to hold on to a full oxygen molecule and wait for 4 electrons to come and reduce that one oxygen molecule into 2 waters. So this is the overall process. I hope that makes a little bit more sense. Let's look at this figure to see if it also helps you understand the process a little bit better. What we see is that NADH donates electrons to complex 1, and that we see also that succinate donates electrons to complex 2. Both of these are going to donate electrons to coenzyme Q, and coenzyme Q is then going to donate its electrons to complex 3. From there, they're passed on to cytochrome C, and then cytochrome C donates its electrons to complex 4. Ultimately, complex 4 donates electrons and reduces oxygen and makes water. Let's come back and review the learning objectives here. I want you to be able to describe the purpose of the electron transport chain, its substrates, products, cellular localization, and tissue distribution. There's a few other objectives that we will be able to get a handle on when we cover the second half of Chapter 20. So that's it for now. Happy studying.